There are creatures on this planet so perfectly engineered in years, and what the answers to that question mean for the future of this ancient lineage. This is part 2 of the dragonfly's story. It is a story that grows more urgent, more complex, and more extraordinary the deeper you go. The adult dragonfly's life begins not with flight, but with waiting. We left the emerging adult in part 1 at the moment of emergence, perched on its plant stem, wings freshly expanded, body hardening in the morning air, entirely vulnerable. Now we follow it through the hours and days that follow, as it transitions from a water-breathing nymph into one of the most accomplished aerial hunters in the history of life. The first hours after emergence are a time of physiological transformation on a scale that is difficult to fully appreciate. Every major system in the dragonfly's body is changing simultaneously. The respiratory system transitions from aquatic to aerial gas exchange. The flight muscles, which were present but undeveloped in the nymph, begin their rapid maturation. They need to reach both structural completeness and the high metabolic capacity required to power sustained flight before the animal can hunt effectively. The visual system, already impressive in the nymph, undergoes its final developmental steps to reach the extraordinary resolution of the adult eye. And the exoskeleton, the cuticle that was soft and flexible during emergence, hardens progressively through a process of sclerotization, in which cross-linking of chitin molecules and the deposition of pigments transforms the pale, soft, newly emerged adult into the hard, brightly colored, structurally robust organism capable of withstanding the physical demands of fast flight. The coloration that develops during this hardening process is one of the most visually striking aspects of the dragonfly's biology, and it has fascinated both naturalists and artists for centuries. The colors of adult dragonflies arise through multiple mechanisms. Some colors are produced by pigments, chemical compounds that absorb certain wavelengths of light and reflect others, producing the reds, yellows, and browns that are characteristic of many skimmer species. Other colors are structural, produced not by pigment, but by the physical interaction of light with nanoscale structures in the cuticle, generating the iridescent blues, greens, and purples that make species like the large blue darner or the various metallic-bodied emeralds so visually spectacular. Structural coloration, which works by the same physical principles as the colors of a soap bubble or a piece of mother of pearl, produces colors that change with viewing angle and that can be extraordinarily pure and saturated, more intense in many cases than any pigment color could produce. The blue of a male blue-tailed damselfly, the metallic green of an emerald dragonfly, the iridescent purple of certain tropical species. These are structural colors, and they are produced by layers of material in the cuticle whose thickness and refractive index are calibrated by evolution to produce specific optical effects. But why are dragonflies colored at all? What evolutionary purpose does the elaborate coloration of adult dragonflies serve? The answer is multifaceted, and different aspects of the coloration serve different functions. Sexual signaling is clearly one function. In most dragonfly species, males are more brightly colored than females, and the male's coloration serves as a signal to both potential mates and rival males. Females of many species exercise mate choice partly based on male coloration, and the brightness and extent of male color patches has been shown in experimental studies to influence mating success. Territorial signaling is closely related. Male dragonflies of most species are intensely territorial during the breeding season, defending patches of suitable oviposition habitat, the stretches of water where females will lay eggs from rival males. A male is a visible territorial signal. His presence at a patch of water, conspicuously perching or patrolling in his full adult coloration, advertises his occupancy and may deter some rivals without requiring the energy expenditure of direct physical confrontation. Thermoregulation is another function of coloration in some species. Dark colors absorb more solar radiation, warming the animal faster in cool conditions. Some dragonfly species adjust their body posture, the angle at which they hold their abdomen relative to the sun, to maximize or minimize solar absorption depending on their thermal state. The famous obelisk posture, in which certain dragonflies point their abdomens directly toward the sun, minimizing the cross-sectional area exposed to solar radiation, is a behavioral thermoregulation strategy that reduces overheating in hot conditions. These animals are actively managing their body temperature through a combination of behavioral and physiological mechanisms that are, for an insect, surprisingly sophisticated. The breeding season is the context in which most human observation of adult dragonflies occurs, and it is one of the most behaviorally rich periods in the animal's life. The water's edge from late spring through summer is a theater of constant activity, patrolling males, hovering females, aerial pursuits, territorial disputes, mating flights, and egg-laying, and understanding what is happening requires knowledge of the evolutionary pressures that have shaped these behaviors. Male dragonflies typically establish territories at the water's edge in the weeks following their emergence. The territory is not a general-purpose home range in the way that the territories of many vertebrates are understood. It is a specific, bounded patch of suitable oviposition habitat, a section of water margin with the right combination of water depth, vegetation structure, substrate type, and sunlight exposure to attract females seeking sites to lay eggs. The male defends this patch not because it contains food or shelter for him. His feeding territory is typically quite different from his breeding territory, but because controlling this patch gives him preferential access to the females who will visit it. The intensity of competition for good oviposition territories is fierce. In many species, the most productive territories, those with the best combination of characteristics that females prefer, attract both the best males and the highest density of competing males. The result is a continuous cycle of encounter and displacement, with territory holders being challenged by wandering males dozens or hundreds of times per day. A male who loses his territory loses access to mating opportunities. A male who successfully holds a high-quality territory during peak female activity can mate many times per day during his tenure. The competitive interactions between males take various forms depending on the species and the nature of the territory. In some species, competitions are largely aerial. The territory holder chases an intruder in a fast, spiraling pursuit flight that may last for several seconds before the loser disengages and retreats. In others, competitions involve prolonged hovering face-offs at the territory boundary, with both males maintaining close proximity while executing hovering flight of extraordinary precision. In some species, physical contact occurs, with males grappling in flight in ways that can be difficult to resolve even with high-speed video. The outcome of these competitions is not determined purely by size or age. Residency has a powerful effect. In experimental studies where territory holders were removed and replaced by new individuals, the new holders quickly became competitive, and when original holders were reintroduced, they often initially lost to the established new holder despite being the same species and similar in size. There appears to be a psychological component to territorial competition in dragonflies, a holder advantage that goes beyond mere physical assessment and reflects something like the motivation asymmetry observed in human economic experiments, where people consistently value losses more than equivalent gains. The female dragonfly navigates this territorial landscape on her own terms. Her choices which territories to visit, which males to mate with, where precisely to lay eggs, drive the selection pressure on all of the male behaviors described above. If females preferred to lay eggs in poor quality sites, or if they mated indiscriminately with whatever male they encountered, male territorial competition over high-quality sites would not be adaptive. It is the female's discrimination, her choosiness in assessing oviposition sites and mates, that makes male competition over these sites evolutionarily meaningful. Female mate choice in dragonflies is an active area of research, and the picture that has emerged is considerably more complex than the simple model in which females passively accept the male who controls the territory they happen to visit. Females assess males through multiple signals simultaneously: coloration, flight performance, the quality of the territory itself, and possibly chemical cues. They can and do reject males they have been mounted by, interrupting mating before sperm transfer is complete. They engage in post-copulatory cryptic mate choice, influencing the outcome of sperm competition after mating through the selective use or displacement of stored sperm. And they exercise considerable independence in their egg-laying decisions, often departing from the territory to lay eggs in a site of their own choosing. The mating system of dragonflies is one of the most studied in entomology, partly because it is complex and partly because it is readily observable at the water's edge with basic equipment. The mechanics of dragonfly mating are anatomically unusual in ways that have attracted considerable scientific and popular attention. Male dragonflies have a secondary genitalia arrangement that is unique in the insect world. The testes and the sperm duct open near the 9th abdominal segment toward the end of the abdomen, but the organs used to transfer sperm to the female are located on the 2nd and 3rd abdominal segments near the front of the abdomen. Before mating, the male transfers sperm from the primary opening at the end of his abdomen to the secondary genitalia at the front, a process called sperm priming or secondary genitalia charging, which requires the male to curl his abdomen forward in a manner visible even to casual observers at the water's edge. The female's reproductive opening is at the end of her abdomen, and during mating, she curves her abdomen forward to bring her reproductive tract into contact with the male's secondary genitalia, while the male grips her behind the head with claspers at the end of his abdomen. The resulting posture, with male gripping female at the head and female curling forward to contact male secondary genitalia, forms what entomologists call the mating wheel or copulation wheel, one of the most characteristic and visually recognizable behaviors at any dragonfly-rich water body. The duration of mating varies enormously among species and among mating events within species, from a few seconds to several hours, and this variation reflects the complexities of sperm competition. When a female mates with multiple males, the sperm of the most recent male has a significant advantage in fertilizing eggs, because the male's secondary genitalia in many species are equipped with structures capable of displacing or removing sperm stored from previous matings before the male deposits his own. This sperm competition dynamic creates a selection pressure for males to prolong mating. Longer matings allow more complete displacement of rival sperm, and a corresponding pressure for females to either resist prolonged mating with males of lower quality or to engage in post-mating behaviors that partially compensate for sperm displacement. The evolutionary dynamics of sperm competition are reflected in remarkable anatomical detail. The sperm displacement structures in male dragonfly secondary genitalia, described by researchers who first characterized them as resembling cleaning brushes, inflatable sacs, or interlocking combs, depending on the species, are precisely calibrated to the anatomy of the female sperm storage organ in the same species. The fit between male sperm displacement apparatus and female sperm storage anatomy is species-specific, suggesting that the two have co-evolved over long periods of antagonistic selection, a biological arms race in which female anatomy evolves to resist sperm displacement and male anatomy evolves to overcome resistance. This kind of coevolutionary arms race between male and female reproductive anatomy is found in other insect groups and has been documented in various vertebrate lineages as well. But in dragonflies, the accessibility of mating behavior for observation and the relative ease of collecting individuals for anatomical study have allowed the phenomenon to be documented with unusual completeness. The dragonfly mating system has become one of the key model systems for the study of sexual conflict, the evolutionary tension between the reproductive interests of males and females in the broader animal behavior literature. After mating, the female faces the critical task of selecting an oviposition site and depositing her eggs. This choice is among the most consequential she will make. The site she selects will determine the environment in which her offspring develop, and the quality of that environment will influence the survival, growth rate, and ultimate reproductive success of the next generation. Oviposition site selection in dragonflies is a complex, multi-stage decision process that integrates information from multiple sensory channels. Visual assessment is primary. Females assess the surface characteristics of the water body, color, reflectance, the presence of emergent vegetation, the structure of the immediate water margin, depth gradient, substrate type, vegetative cover, and the broader landscape context, proximity to suitable adult habitat, absence of obvious threats. Chemical assessment also plays a role in some species. Females can detect chemical cues from the water that provide information about its quality, prey availability for nymphs, and possibly the presence of other dragonfly eggs or nymphs. One of the most striking and well-documented aspects of oviposition site selection is the polarized light trap, a phenomenon that causes serious problems for dragonfly populations in modern, human-altered landscapes. Dragonflies use the polarization of reflected light as a cue for identifying water surfaces. Water reflects polarized light in a characteristic way, specifically, horizontally polarized light, and dragonflies are sensitive to the polarization plane of light in a way that allows them to distinguish water surfaces from other bright, flat surfaces. This was an extraordinarily reliable cue in the evolutionary environment of the dragonfly, because essentially, the only surfaces that produced strong horizontal polarization in that environment or actual water surfaces. But some artificial surfaces also produce strong horizontal polarization. The shiny black surfaces of solar panels, black plastic sheeting, dark asphalt roads, certain types of windows, and oil slicks on pavement all reflect horizontally polarized light that is, to a dragonfly's visual system, indistinguishable from the reflection of a genuine water surface. Dragonflies are attracted to these surfaces and attempt to mate and oviposit on them. Females deposit eggs on asphalt roads, on solar panels, on plastic sheeting, in an entirely futile reproductive effort that benefits neither the eggs nor the female, but wastes her remaining reproductive energy. This ecological trap, a situation in which an organism's evolved detection system leads it toward an artificial stimulus that mimics the target stimulus but provides no biological benefit, is one of the clearest examples of how rapid human alteration of the landscape can disrupt the evolved sensory systems of animals whose evolution occurred in a pre-industrial world. The design of solar panels and other reflective surfaces to be less attractive to dragonflies, for instance, by adding cross-polarizing grid patterns that break up the uniform polarization signal, is an active area of applied research in conservation biology. It represents a case in which understanding the sensory biology of a wildlife species can directly inform the design of human infrastructure in ways that reduce unintended ecological harm. The act of egg deposition itself varies enormously among dragonfly species, reflecting the diversity of freshwater habitats they exploit and the diversity of nymphal requirements across species. This diversity in oviposition behavior is one of the clearest expressions of the ecological radiation of the Odonata, the way a single fundamental body plan has been adapted to exploit an extraordinarily wide range of freshwater environments. Endophytic oviposition is the most behaviorally elaborate egg-laying strategy found in many hawker dragonflies and many damselflies. In this strategy, the female uses her ovipositor, a blade-like structure derived from modified abdominal appendages, to cut slits into aquatic or waterside plant tissue and insert individual eggs into the plant material. The eggs develop protected within the plant tissue, safe from the desiccation that would kill eggs exposed on open water, and in some cases within the chemical environment of the plant, which may provide additional protection against pathogens and parasites. The female can spend long periods at a single stem, methodically working her way up or down the plant, cutting and inserting with a precision that is remarkable to observe. Exophytic oviposition involves depositing eggs directly into or onto the water, or onto substrate at the water margin. Some species hover over the water, dipping the tip of the abdomen repeatedly to the surface with each contact leaving a small cluster of eggs on the water film. Some species land on substrate at the water's edge and drag the tip of the abdomen across the wet surface, leaving a trail of eggs in the thin film of moisture. Some species simply hover and release eggs into the water from above, allowing them to sink to the substrate. In all of these strategies, the eggs are immediately exposed to the aquatic environment, and their survival depends on the quality of that environment. The eggs of some species can survive temporary desiccation if the water body dries out, a critically important adaptation for species that exploit temporary ponds and seasonal wetlands. These drought-resistant eggs enter a dormant state, metabolically quiescent, capable of surviving for weeks or months in dry substrate, and then rehydrating and resuming development when water returns. This adaptation gives some species access to temporary habitats that would otherwise be unavailable, and it provides a form of temporal insurance against environmental variability, the ability to spread reproductive risk across wet and dry years by having some eggs survive the dry period to hatch when conditions improve. In some species, the eggs are not simply deposited and left, but are encased in substances that modify their immediate microenvironment. Gelatinous matrices surround eggs in many species, providing moisture retention, physical protection, and in some cases chemical defense. The chemistry of these matrices is another area of active research, with some studies suggesting that the gel contains antimicrobial compounds that protect the developing embryo against pathogens in the aquatic environment. The hatching of the dragonfly egg initiates the nymphal stage, and the first instar nymph that emerges from the egg is, by almost any standard, an astonishing thing. It is tiny, often barely visible to the naked eye, perhaps a millimeter or two in total length. It is nevertheless fully equipped with functional compound eyes, functioning legs, and critically, a functional labium already capable of the hydraulic strike mechanism that it will use throughout its nymphal life. The predator is ready from the first moment of its aquatic existence. The first instar nymph typically hatches as what is called a pronymph, a very brief non-feeding stage that transitions rapidly to the first true instar. In some species, the pronymph stage lasts only minutes. In others, it may persist for a few hours. In a very few species, the pronymph is enclosed in a specialized cuticle that must be shed before the true first instar can begin feeding. The evolutionary significance of the pronymph stage is debated. It may represent a remnant of a more extended intermediate stage in ancestral Odonata, or it may serve a specific function in the very earliest phase of aquatic life that is not yet fully understood. The progression from first instar to final instar is governed by a hormonal cascade in which the balance between juvenile hormone, which suppresses development toward the adult form, and ecdysone, which triggers molting, shifts gradually across instars. In the early instars, juvenile hormone is dominant, maintaining the nymphal form through each successive molt while allowing growth. As the nymph enters its later instars, the sensitivity to ecdysone increases and the production of juvenile hormone declines, allowing the progressive development of adult structures. The compound eyes expand, the wing pads become prominent, the reproductive organs develop, and the brain undergoes further elaboration toward the adult configuration. The final instar is marked by a particularly dramatic internal change. The tracheal system, which in the nymph delivers oxygen supplemented by the rectal gill system, prepares for the transition to a purely aerial respiratory mode. The spiracles, the openings through which air will enter and exit the adult's tracheal system, mature and become functional. The flight muscles undergo a final phase of growth and differentiation. And the neuroendocrine system generates the surge of signals that will eventually trigger the emergence behavior, the climb out of the water, the splitting of the thoracic cuticle, and the extraordinary transformation of adult emergence. Now we must return to the globe skimmer and trace its migration in the detail it deserves, because this story is not simply a curiosity about insect biogeography. It is a window into the global connectivity of freshwater ecosystems, the relationship between atmospheric circulation and animal movement, and the extraordinary ways in which evolution can build behavioral systems that operate at scales that seem impossible for organisms of the size involved. The globe skimmer Pantala flavescens belongs to the family Libelulidae, the skimmers, which is itself the largest dragonfly family, containing more species than all other dragonfly families combined. Within the skimmers, Pantala is unusual in multiple respects. It is larger than many skimmers, with a wingspan of approximately 70 to 80 millimeters, not enormous for a dragonfly but substantial. Its wings are notably broad, with a wing area relative to body mass that is the highest known among dragonflies, a characteristic that is directly relevant to its migratory capabilities, as we will discuss. Its coloration is a warm, translucent amber yellow that gives it both its scientific name, flavescens, yellowish, and a distinctive appearance in flight, where its wings catch the light in a way that makes large aggregations unmistakable. The globe skimmer has a strong preference for temporary water bodies, seasonal rain pools, flooded fields, temporary ponds over permanent lakes and rivers. This preference is directly linked to its migratory lifestyle because temporary water bodies form where seasonal rainfall is abundant and the globe skimmer follows the rainfall. Its migration is, at its most fundamental level, a pursuit of the rain, of the temporary freshwater habitats that form in the wake of monsoon precipitation and that provide the oviposition sites and nymphal development environments the species requires. The monsoon systems that the globe skimmer exploits are among the largest and most powerful atmospheric phenomena on Earth. The Indian Ocean monsoon, more properly a complex of related monsoon systems, drives seasonal precipitation across a vast region spanning from East Africa to South and Southeast Asia, with the wind direction reversing between summer and winter hemispheres. The northeast monsoon moves from Asia toward Africa during the northern winter, while the southwest monsoon flows in the opposite direction during the northern summer. Within these large-scale wind patterns at altitudes of several hundred to over 1,000 meters above the surface, the globe skimmer rides the winds on journeys that individual animals cannot complete in a single generation. The full circuit of the globe skimmer's migration, as reconstructed from isotope data, direct observation, and atmospheric modeling, runs roughly as follows. Populations develop in South Asia, particularly the Indian subcontinent, Sri Lanka, and adjacent regions, during the period of the southwest monsoon, which brings rainfall and creates the temporary pools the species requires. As the monsoon retreats and the temporary pools dry, the adult dragonflies rise to altitude and are carried southwestward by the retreating monsoon winds across the Arabian Sea and toward East Africa. This crossing of the open ocean, a distance of several thousand kilometers, is completed using the tailwinds of the monsoon system, without which it would be impossible. In East Africa, the arriving globe skimmers find temporary pools formed by local rainfall and reproduce. Their offspring, the nymphs, develop rapidly in the warm, shallow temporary pools, and the adults that emerge face a different seasonal wind pattern that carries them onward. Some populations move to southern Africa, following rainfall southward as the austral summer approaches. Others remain in East Africa and reproduce again before the seasonal winds shift once more. Eventually, populations are carried back toward the Indian Ocean islands and onward toward South and Southeast Asia, completing the circuit across generations. The logistics of this migration are extraordinary when examined in detail. The open ocean crossing from the Indian subcontinent to East Africa covers approximately 4,000 kilometers over water with no land in between, no opportunities for rest, no fresh water to drink, no prey to catch. The globe skimmer must sustain flight for this entire distance, fueled entirely by the fat reserves it accumulates before departure. Its broad wings, the highest wing-loading ratio among dragonflies, are specifically adapted for sustained, efficient soaring and gliding, rather than the rapid, highly maneuverable hunting flight of forest-dwelling species. It can exploit rising air currents, thermal updrafts, and the consistent push of the monsoon winds to minimize the energy cost of the crossing. Studies of globe skimmer energetics have attempted to calculate whether the fat reserves of a departing individual are actually sufficient to fuel the ocean crossing. The calculations are tight, the energy budget barely works out, with little margin for error. This means that the migration is possible only under favorable conditions, with consistent tailwinds, the absence of storms that would force extended active flapping, and perhaps the occasional exploitation of thermal updrafts that reduce the cost of maintaining altitude. The monsoon winds are not perfectly consistent, they fluctuate in direction and intensity, and it is likely that not all individuals that begin the ocean crossing complete it successfully. Losses during the crossing may be significant, but those that do complete the crossing arrive in East Africa and participate in reproduction, contributing to the next generation that will eventually make the return journey. The multi-generational nature of the migration means that the losses of any one generation are compensated by the production of the next. The population is the migratory unit, not the individual, and the population's circuit across the Indian Ocean is maintained by the collective behavior of millions of individuals across multiple generations. The isotope analysis that confirmed the transoceanic migration of the globe skimmer represents a methodological achievement worth examining in its own right, because it illustrates how modern scientific tools can resolve biological questions that direct observation cannot answer. The stable isotope ratios of hydrogen, specifically the ratio of deuterium, a heavy isotope of hydrogen, to the common light isotope, in precipitation vary systematically across the globe. This variation is driven by the progressive depletion of heavy isotopes as water vapor moves inland from the ocean and undergoes successive rounds of condensation and precipitation. Coastal rainfall has a higher proportion of deuterium than interior rainfall. Rainfall that has traveled a long distance from its oceanic source is progressively lighter in its isotopic composition. Additionally, temperature affects isotopic fractionation during evaporation and condensation, meaning that rainfall at different latitudes and elevations has characteristic isotopic signatures. The freshwater that dragonfly nymphs drink and metabolize during their development is incorporated into their body tissues, including the chitin of the cuticle. When the nymph molts into the adult form during emergence, the new adult cuticle is synthesized from materials that carry the isotopic signature of the water in which the nymph developed. The wing chitin deposited during this final molt is a stable, chemically resistant material that retains its isotopic composition through the adult's life, and indeed for long periods after death. Researchers can extract chitin from the wings of preserved dragonfly specimens and measure its deuterium to hydrogen ratio with precision using isotope ratio mass spectrometry. The resulting isotopic value can then be compared to maps of precipitation isotope ratios across the globe, maps that have been constructed from measurements at hundreds of precipitation collection stations over many years. By finding the geographic region whose precipitation isotopic signature best matches the isotopic value in the wing chitin, researchers can identify where the dragonfly grew up. When this analysis was applied to globe skimmers collected in East Africa and the Maldives, locations where local rainfall has a characteristic isotopic signature, the chitin isotope values did not match those of local precipitation. They matched the isotopic signatures of precipitation from the Indian subcontinent. This mismatch between the isotopic signature of the collection location and the isotopic signature encoded in the wings of the collected individuals is direct, quantitative evidence that these dragonflies did not develop where they were collected. They flew there, from thousands of kilometers away, carrying the chemistry of their birthplace in their wings. This result has been replicated across multiple studies, with multiple collections from multiple locations around the Indian Ocean basin, using both hydrogen isotope ratios and, in some studies, oxygen isotope ratios as an independent confirmation. The consistency of the results across studies and methods gives the conclusion a robustness that goes well beyond what a single study could provide. The globe skimmer's transoceanic migration is not a hypothesis. It is a demonstrated fact, confirmed by multiple independent lines of evidence, and it stands as one of the most remarkable biogeographic phenomena documented in the insect world. The globe skimmer is not the only migratory dragonfly, though it is the most extensively studied and the most dramatically migratory. Long-distance movements in dragonflies appear to be more widespread than previously appreciated, and new observations, facilitated by citizen science reporting platforms, radar studies of high-altitude insect movement, and expanding isotope analysis programs, are continuing to expand the known scope of odonatin migration. In various parts of the world, large aggregations of dragonflies appear seasonally at locations far from known breeding populations, move in consistent directions, and disappear as seasonal conditions change. Some species of darner dragonflies undertake significant north-south movements in response to seasonal temperature changes, with populations moving southward in autumn and northward in spring in a pattern that is functionally analogous to the movements of migratory birds in the same regions. These movements are facilitated by physiological pre-adaptations that are found across the dragonfly order more broadly, not just in confirmed migrants. The ability of dragonflies to store large fat reserves, their capacity for sustained flight over long periods, their use of thermals and wind currents to minimize the energetic cost of movement. All of these capabilities are present to varying degrees across the Odonata, and in migratory species they are present in their most extreme expressions. This suggests that migration may have evolved multiple times independently across the dragonfly family, with the underlying physiological capacity for sustained movement being recruited into migratory behavior whenever ecological conditions, particularly the distribution of suitable freshwater habitats across a landscape with seasonal rainfall variation, favored it. The ecological consequences of dragonfly migration extend beyond the movement of individuals across landscapes. Migratory dragonflies carry genetic material between populations, as already noted for the globe skimmer. They also carry parasites, the mites and gregorines and other organisms that parasitize dragonflies in one region can potentially be transported to new regions by migrating hosts. And the arrival of large aggregations of migratory dragonflies in a new location has trophic effects on the local ecosystem. Their predation on local insects during stopover or arrival, and their own use as prey by local predators, integrate them into food webs far from their home populations. The most spectacular aspect of dragonfly aggregations, whether migratory or local, is their sheer scale. When globe skimmers arrive on the Maldivian islands from the Indian subcontinent, the numbers can be almost incomprehensible. Observations record millions of individuals arriving over the course of a few days, darkening the sky over the low-lying atolls and covering every available surface. Local people have developed cultural responses to these arrivals, festivals, harvesting traditions, dragonflies are eaten in some island cultures, and predictive folk knowledge about the timing and scale of arrivals based on wind and weather. The ecological importance of these mass arrivals, both for the dragonflies themselves, who face a dramatic shortage of suitable oviposition habitat on coral atolls, and for local predators, who enjoy a dramatic temporary food bonanza, is an understudied area with considerable potential for future research. The neuroscience of the adult dragonfly's visual system deserves its own extended treatment, because what researchers have discovered in the neural circuits of the dragonfly brain represents some of the most elegant and informative biological computation known to science. This is not simply a matter of curiosity about an insect's capabilities. The dragonfly visual system has become a model for understanding how nervous systems can perform complex computations with minimal neural tissue, a question with profound implications for our understanding of neural computation in all animals, including ourselves. The visual information from the compound eye enters the brain through a series of visual processing layers: the lamina, the medulla, and the lobula complex, each performing progressively more complex operations on the raw visual signal. In the lamina, the basic signals from individual photoreceptors are processed into contrast and motion signals. In the medulla, directional motion information is extracted. The system begins to distinguish what is moving from what is stationary and in what direction movement is occurring. By the time visual information reaches the lobula complex, neurons are responding selectively to specific classes of visual objects and motions. Among the neurons in the dragonfly lobula complex, one class has attracted extraordinary scientific attention: the small target motion detector neurons, or STMDs. These neurons, first described in the 1970s and studied in increasing detail since, are among the most precisely tuned neurons in any nervous system. They respond selectively to small, dark targets moving against a complex, cluttered background, exactly the visual signature of a small insect flying through the scene. They respond to this stimulus, and crucially, they are relatively unresponsive to large field motion, such as the motion of the entire visual field that accompanies the dragonfly's own flight, or the motion of large objects like leaves moving in the wind. This selectivity for small targets in the presence of large field motion is not a trivial computational achievement. Any visual system that attempted simply to detect motion would be overwhelmed by the motion of the entire scene during flight. The STMD neurons somehow subtract the large field motion, which represents the observer's own movement, and extract the signal of the small, independently moving target from the background of self-motion. How they do this computationally is still an area of active investigation, but the neural circuit mechanisms involved are beginning to be understood, and they involve a specific type of neural interaction between the cells that detect small field motion and cells that estimate the large field motion pattern. The individual STMD neurons respond to targets that are very small as a single facet of the compound eye, subtending a visual angle of less than a degree and moving at high speed. They can track a target through a visually complex background, the irregular, high-contrast pattern of sunlit leaves that constitutes the most difficult background for any target detection system, and they do so with a reliability that machine vision systems have struggled to match. When researchers project small, moving targets against complex moving backgrounds inside laboratory chambers and record the electrical activity of individual STMD neurons, the selectivity and reliability of the responses are consistently impressive. The STMD system does not work in isolation. It feeds into a larger system of neurons that perform the predictive trajectory calculation described in Part 1, the computation of where the prey will be at the moment of interception. Neurons downstream of the STMDs have been identified that integrate target position information across time, effectively computing velocity and extrapolating future position. The entire neural circuit, from the photoreceptors of the compound eye to the motor neurons that control the flight muscles, constitutes a remarkably complete and efficient solution to the problem of detecting, tracking, and intercepting a small, fast-moving target in a complex visual environment. What makes the dragonfly system especially valuable to neuroscience is its accessibility. The neurons are large enough to record individually with metal electrodes. The animal can be prepared for recording in ways that allow natural visual stimulation while maintaining neural access, and the behavior, prey capture in flight, can be studied in parallel with neural recording, allowing researchers to directly correlate neural activity with behavioral outcomes. This combination of experimental accessibility and behavioral relevance has made the dragonfly visual system one of the most productive model systems in sensory neuroscience over the past several decades. The 80% allocation of adult dragonfly brain tissue to visual processing is a number that deserves careful contextualizing, because it is easy to state and easy to hear without fully grasping its implications. To allocate 80% of a biological organ, an organ that is energetically expensive to grow and maintain, that imposes weight penalties on a flying animal, and that requires a blood supply and oxygen delivery system proportional to its size, to a single sense is a statement about the evolutionary environment in which that allocation was optimized. It means that for an adult dragonfly, vision is survival. It means that the vast majority of the information the dragonfly needs to make all of the decisions that matter to its survival and reproduction, where to go, what to eat, who to mate with, which territories to enter or avoid, where to lay eggs, is available in visual form and is extracted from visual information by the enlarged visual brain. The other senses, mechanosensory, olfactory, auditory, are present and functional, but they are supplementary. The world that matters to the dragonfly is the world that can be seen. This has a curious philosophical implication. The dragonfly and the human share the same physical world, the same ponds, the same afternoon light, the same freshwater ecosystems. But the subjective experiences of that world, if we are willing to grant that the dragonfly has anything analogous to subjective experience, are radically different. The human world is multisensory in a genuinely balanced way. We process sound and touch and smell alongside vision, and these other senses contribute substantially to our awareness at every moment. The dragonfly's world is visual to a degree that is almost exclusive. It lives in a world of light and motion, shape and color, targets and backgrounds, polarization patterns and ultraviolet signals invisible to human eyes, in a panoramic visual field that covers nearly the entire sphere of space around its body. What does it mean to be in that visual world? What does a pond look like from inside the visual system of a creature that has dedicated 80% of its brain to seeing it? These are questions that science cannot directly answer. The neural correlates of subjective experience remain one of the deepest unsolved problems in all of science, and the dragonfly is not exempt from this uncertainty. But they are questions worth asking, because they remind us that the familiar animals of our familiar landscapes are experiencing worlds that are not familiar at all, that the pond at the end of a summer afternoon is, for the dragonfly hovering over it, a place of extraordinary richness and complexity that our own senses cannot fully access. The relationship between the dragonfly and human culture is one of the most extensive and cross-cultural of any insect. The dragonfly appears in the art, mythology, and cultural practice of societies on every inhabited continent and across the span of recorded human history. This cultural ubiquity is itself a testament to the dragonfly's biological success. You cannot develop cultural associations with an organism you never encounter, and the dragonfly's presence at virtually every freshwater habitat in the world has made it a companion of human life since our species first settled near water, which is to say, always. In ancient Egypt, the dragonfly appears in tomb paintings and as a hieroglyphic element in written language, associated with resurrection and the liminal boundary between the living world and the afterlife. In medieval Europe, the dragonfly was associated with supernatural forces. The folk name adder's needle, common in parts of England, reflected a belief that dragonflies were associated with snakes. In some Scandinavian traditions, dragonflies were connected to the devil, a cultural valuation that has no obvious biological basis, but reflects the ancient human tendency to project human moral categories onto natural phenomena that are striking and not fully understood. In Japan, the cultural relationship with the dragonfly is perhaps the deepest and most articulate of any. The dragonfly appears in Japanese poetry, art, and mythology going back to at least the Nara period in the 8th century. It is associated with the autumn season and with the spirit of departed ancestors returning during the Aben festival. The name for Japan in one ancient literary tradition, Akitsushima, the island of the dragonfly, reflects a mythological narrative in which the Emperor Jimmu, viewing his realm from a high peak, compared its shape to two dragonflies mating. The samurai class adopted the dragonfly as a symbol of martial valor. The dragonfly, like the ideal warrior, never retreats but only moves forward, a piece of folk natural history that is not biologically accurate but captures something true about the dragonfly's assertive, direct flight style. Dragonfly motifs appear on samurai helmets, sword guards, and armor, and the cultural prestige of the dragonfly image has persisted into modern Japanese design and aesthetics. In the cultures of many indigenous peoples of various continents, the dragonfly holds significant spiritual or cosmological meaning. In some traditions, the dragonfly is associated with water and rain, an association that is ecologically accurate given the dragonfly's dependence on freshwater habitats. In others, it is a symbol of transformation, again reflecting something biologically real. The metamorphosis from aquatic nymph to aerial adult is one of the most dramatic biological transformations visible to the unaided human eye. In still others, the dragonfly serves as a messenger between the human world and the spirit world, occupying a liminal position between elements, water and air, that resonates with its actual biology. The common thread across this extraordinary cultural diversity is transformation. The dragonfly, across virtually every culture that has developed associations with it, is linked to change, transition, and the crossing of boundaries. This cultural resonance is not accidental. It reflects a genuine biological reality. The dragonfly truly does cross the boundary between two worlds, spending its life first in the hidden world of water and then in the visible world of air. Its transformation from nymph to adult is real, dramatic, and visible to anyone who has observed it. The cultural associations that humans have built around this transformation are one of the most direct translations of biological reality into human meaning. The evolutionary history of the Odonata through the 5 mass extinction events of the Phanerozoic deserves examination in greater detail than was possible in Part 1, because each extinction event presents a different challenge, and the dragonfly's survival of each illuminates different aspects of its resilience. The first major extinction event relevant to the dragonfly lineage is the late Devonian extinction, which began approximately 375 million years ago, before the first recognizable dragonfly ancestors appear in the fossil record, but during the period when the Odonatan lineage was diverging from its closest relatives. The late Devonian extinction primarily affected marine organisms, particularly the reef-building organisms and the open-water vertebrates and invertebrates of the productive late Devonian seas. Terrestrial and freshwater ecosystems were affected but less severely, and the aquatic arthropod lineages that were becoming the Odonatan ancestors apparently navigated this extinction with relatively modest losses. The Permian-Triassic extinction, the Great Dying, approximately 252 million years ago, is the relevant test case for understanding dragonfly resilience under maximum stress. This was the most catastrophic biological crisis in the history of complex animal life, and no lineage was untouched by it. The mechanisms of the extinction, rapid warming driven by Siberian traps volcanism, oceanic anoxia, acidification of both freshwater and marine systems, disruption of terrestrial food webs were directly relevant to dragonfly biology. Freshwater systems warmed and deoxygenated. The aquatic insects and invertebrates on which nymphs depended were decimated. The terrestrial insect prey of adult dragonflies was severely depleted. The physical structure of freshwater habitats was disrupted by the destabilization of terrestrial vegetation that fed them organic material and regulated their hydrology. That any dragonfly lineages survived this event is remarkable. That the entire Odonatan order survived, and that the post-Triassic fauna included multiple dragonfly families with representatives clearly ancestral to modern groups, is extraordinary. The most likely explanation is a combination of factors. The breadth of the nymph's dietary flexibility, able to survive on whatever aquatic invertebrates persisted, the ability of some populations to retreat to refugia, areas where freshwater conditions remained adequate through the worst of the crisis, and the capacity for egg dormancy that allowed some populations to sit out extended dry or unfavorable periods in the egg stage. The Triassic-Jurassic extinction, approximately 201 million years ago, was less severe than the Permian-Triassic but still substantial, eliminating perhaps 70 to 80% of species, particularly large terrestrial vertebrates. The dragonfly passed through this event with apparent equanimity. The Jurassic fossil record of dragonflies is extensive and diverse, showing a fauna that had fully recovered from whatever Permian-Triassic losses occurred and had diversified into new forms. The Jurassic dragonfly fauna is documented in exceptional quality from several fossil deposits around the world, including the lithographic limestone of the Solnhofen Formation, the same deposit that produced the famous Archaeopteryx fossils, and equivalent deposits in China, Central Asia, and other regions. The Solnhofen dragonflies are exquisitely preserved, with wing venation preserved in such detail that modern entomologists can assign many of them to specific families with confidence. Examining these fossils, you see animals that are not the griffinflies of the Carboniferous, not the enormous ancestral dragonflies of the Permian, but something recognizably close to the modern fauna, smaller, more diverse, with family-level representatives of groups that lead directly to modern lineages. The end Cretaceous extinction, 66 million years ago, provides a final test. The asteroid impact that ended the Mesozoic era created an immediate crisis of darkness and cold, the so-called impact winter, that lasted months to years, followed by a period of warming as carbon dioxide from the impact and associated fires accumulated in the atmosphere. Freshwater systems experienced temperature shock, disrupted hydrology as the terrestrial vegetation that regulated watersheds was killed, and the collapse of terrestrial food webs that fed organic material into aquatic systems. Yet the dragonfly lineage survived, and the Paleogene fossil record shows dragonflies diversifying rapidly into the post-extinction world, exploiting the ecological opportunities created by the extinction of so many competing and predatory lineages. The pattern across all 5 extinctions is consistent. The dragonfly lineage loses diversity during the crisis, sometimes substantially, but retains enough lineages and enough refugia to repopulate the world when conditions improve. The freshwater anchor, the dietary flexibility of the nymph, the egg dormancy capacity of some species, and the inherent resilience of a body plan that has been optimized across hundreds of millions of years all contribute to this consistent pattern of survival through catastrophe. Understanding the dragonfly's past, we must now confront its present and future. And this requires confronting some deeply uncomfortable realities about the trajectory of freshwater ecosystems globally. The figure introduced in Part 1 bears repeating and expanding. Approximately 35% of the world's wetlands have been lost since 1970. This is not a geological estimate subject to the uncertainties of the fossil record. It is a measured quantity, derived from satellite imagery, land survey data, and carefully assembled global datasets of wetland area and condition. In the span of 50 years, roughly 2 human generations, more than a third of the world's wetlands have been converted, degraded, or eliminated. The causes of this loss are multiple and vary by region, but several drivers are globally significant. Agricultural drainage is the largest single cause of wetland loss in many regions. Converting wetland to farmland has been practiced for millennia, but the pace accelerated dramatically in the 20th century with the mechanization of agriculture and the development of effective drainage technology. The productivity benefits of converting wetland to farmland are real and immediate, while the ecological costs are diffuse, delayed, and distributed, experienced not by the farmer who drains the wetland, but by the downstream communities that depended on the water filtration, flood buffering, and biodiversity support that the wetland provided. Urban expansion has buried countless ponds, marshes, and stream corridors under impervious surfaces in every region of the world where cities have grown. The small ponds and ditches that once dotted agricultural and semi-natural landscapes have been eliminated by land consolidation, straightening and channeling of streams, and the conversion of irregular, water-retaining landscapes into smooth, efficiently drained ones. These small water bodies, individually insignificant in area but collectively enormous in importance, were the habitats of many dragonfly species, and their loss has reduced dragonfly diversity and abundance across vast areas. Water extraction, the pumping of groundwater, and the diversion of surface water for agriculture, urban supply, and industrial use, lowers water tables in ways that drain seasonal wetlands even without any direct physical modification of the wetland itself. A seasonal pond that depends on a high water table to maintain water through the dry season disappears when the water table drops below the pond's base level. No physical intervention in the wetland is required. The pump miles away does the work invisibly, and the pond simply fails to fill in the next wet season and the next, and eventually it becomes just another dry patch of ground. Pollution is another pervasive threat. Agricultural runoff carries excess nutrients, particularly nitrogen and phosphorus from fertilizers, into freshwater systems. These nutrients drive algal growth, and when the algae die and decompose, the microbial decomposition process consumes dissolved oxygen, driving the oxygen levels in the water below the threshold that dragonfly nymphs and most other aquatic life require. This process, called eutrophication, transforms a clear, biodiverse, oxygen-rich water body into a turbid, low-oxygen, species-poor one. The transition can happen relatively quickly, over the course of a few years to decades, and reversing it once it has occurred is considerably more difficult than preventing it. Pesticides represent another category of chemical threat. Insecticides in particular are directly lethal to dragonfly nymphs at concentrations that can occur in agricultural runoff. Neonicotinoid insecticides, which became widely used from the 1990s onward, are highly water-soluble and persistent, and they have been detected in freshwater systems across agricultural landscapes at concentrations that have measurable effects on aquatic invertebrate communities, including dragonfly nymphs. The systemic nature of neonicotinoids, they are taken up by plants and distributed through plant tissues, including pollen and nectar, means that they reach aquatic systems through multiple pathways, including direct runoff, leaching through soil, and atmospheric deposition. Invasive species represent a biological rather than chemical threat, but one that can be equally devastating. The introduction of predatory fish into ponds that previously lacked fish is one of the most consistently damaging ecological interventions in freshwater habitats. Fish-free ponds support high densities of dragonfly nymphs and other large invertebrates because these organisms evolved without fish predation and have not evolved the behavioral and morphological defenses that allow coexistence with fish. When fish are introduced for fishing recreation, or accidentally through the dumping of bait fish, or through flooding events that allow fish from connected waterways to colonize new habitats, they can eliminate dragonfly nymph populations within a few years. The cumulative effect of all of these threats, drainage, urban expansion, water extraction, pollution, invasive species, is a freshwater biodiversity crisis that parallels and in some measures exceeds the better publicized crises in terrestrial and marine habitats. Freshwater species, including dragonflies, are declining at rates that consistently exceed the declines observed in comparable terrestrial species. The global biomass of freshwater insects is declining. The ranges of freshwater species are contracting. Local extinctions are accumulating into regional losses that are themselves accumulating into global losses. Yet the story of the dragonfly's relationship with wetland conservation is not only a story of loss. It is also, in important ways, a story of resilience and recovery, a story that reflects the same deep biological robustness that has carried this lineage through 5 mass extinctions. Wetland restoration, the active work of recreating or rehabilitating degraded freshwater habitats, has been practiced with increasing sophistication over the past several decades, and the results are in many cases genuinely encouraging. Dragonflies are among the fastest and most reliable colonizers of restored wetlands. When a suitable water body is created, even from entirely artificial origins, such as a gravel pit that has been flooded after the end of extraction, dragonfly populations can establish within the first year, and species diversity can reach levels comparable to long-established natural water bodies within a decade. This rapid colonization reflects several aspects of dragonfly biology that we have already examined. The highly mobile adult stage means that even newly created water bodies are quickly discovered and assessed by searching females. The broad dietary tolerance of nymphs means they can exploit the developing invertebrate community of a new water body even before it reaches full ecological maturity. The ability to reproduce successfully in small, isolated water bodies means that metapopulations, networks of small, interacting local populations linked by dispersal, can maintain themselves across landscapes where individual water bodies are small and scattered. The conservation value of small water bodies deserves particular emphasis because it runs counter to an intuition that leads some to undervalue them. A small garden pond, a farm pond, a flooded ditch, a restored gravel pit, none of these is individually impressive as a nature reserve. None of them compares in area or in biodiversity to a large natural lake or an extensive wetland complex. But the cumulative importance of small water bodies distributed across agricultural and suburban landscapes is enormous. In many regions, the majority of the freshwater habitat available to dragonflies consists not of large, protected water bodies, but of numerous small, often human-created or human-modified water bodies scattered across the landscape. Studies that have surveyed dragonfly communities across landscape gradients consistently find that the density and connectivity of water bodies is among the most important predictors of dragonfly diversity. A landscape with many small ponds within dispersal distance of each other supports higher dragonfly diversity than a landscape with fewer, larger water bodies, even when the total water surface area is similar. This is because different species have different habitat requirements, and a mosaic of different small habitats supports a greater variety of species than a single large habitat with homogeneous characteristics. The practical implication of this finding is significant. Conservation of dragonfly communities does not require massive, landscape-scale restoration of ancient wetland complexes, though where such restoration is possible, it should certainly be pursued. It can also be achieved through the creation and management of networks of small water bodies, including water bodies in human-dominated landscapes like farms, gardens, parks, and the margins of roads and railways. Every pond created, every ditch maintained with water, every garden water feature managed without fish, contributes to the network of habitat that dragonfly populations require. The monitoring of dragonfly populations as indicators of freshwater ecosystem health is increasingly recognized as one of the most valuable tools available to conservation managers and freshwater ecologists. Dragonflies have several characteristics that make them exceptionally good indicators. They are sensitive to water quality, as already discussed, making them responsive to pollution and other water quality changes. They are present in the nymphal stage throughout the year, providing a continuous biological record of conditions rather than a seasonal snapshot. They are taxonomically identifiable to species level from both adults and exuvii, and the species compositions of communities reflect the specific characteristics of the water body. And they are large enough to be collected, identified, and counted without specialized equipment that requires significant investment or expertise to use. The exuvia survey, the systematic collection and identification of the shed nymphal cases left on emergent vegetation at the water's edge following adult emergence, is one of the most powerful monitoring tools available. A thorough exuvia survey of a water body during the emergence season, which varies by species and region, provides a precise census of which species emerged from that water body, in what numbers, and over what time period. Because the exuvii are left at the point of emergence, they also provide spatial information about which parts of the water body were used for emergence. And because the exuvii can be identified to species level by examining the specific details of the labium, the eye shape, the abdominal structure, and the overall body form, a careful observer with a hand lens and an identification guide can extract a remarkable amount of information from what are, in essence, empty insect shells. Exuvia surveys have been used to track population changes over time, to assess the colonization of restored wetlands, to detect the impacts of pollution events on dragonfly communities, and to monitor the effects of management interventions on habitat quality. Long-term exuvia datasets from consistently surveyed water bodies now exist for some locations in various parts of the world, and these datasets are providing ecologically important information about how dragonfly communities are responding to the environmental changes of the early 21st century. The dragonfly has survived 300 million years. It has survived ice and fire, impact and volcanism, warming and cooling, the rise and fall of dynasties of organisms that we regard as the most spectacular and successful the Earth has produced. It has done this by being in every essential respect already the answer, already the completed solution to the problem it was asked to solve. It did not need to change because nothing better suggested itself, and nothing better suggested itself because there was nothing better to be. But there is something new in the world that the dragonfly has not encountered before. Not in any previous extinction event, not in any previous period of climate disruption, not across any of the 300 million years of its existence. What is new is speed. The transformation of the freshwater world that the dragonfly depends upon is happening at a pace that has no geological precedent. The Permian-Triassic extinction unfolded over tens of thousands of years. The post-Cretaceous impact winter resolved over years to decades. Even the most rapid past climate shifts, driven by orbital cycles, occurred over thousands of years, time enough for populations to track changing habitat boundaries, for range shifts to compensate for local losses, for the metapopulation networks that sustain species through landscape change to function and adjust. What is happening to wetlands now is happening over decades. Within a single human lifetime, well within living memory, a third of the world's wetlands have disappeared. This pace of change is faster than the pace at which dragonfly populations can adapt through evolutionary change. It may, in some regions and for some species, be faster than the pace at which populations can track the moving boundaries of suitable habitat. And it is globally comprehensive in a way that past extinction events were not, affecting not one region while leaving others as refugia, but occurring simultaneously across the planet, driven by globally distributed human economic activity. This does not mean that the dragonfly is doomed. It has survived things that seem, in retrospect, unsurvivable. It will almost certainly survive the 21st century in some form, in some habitats, in some populations. The question is not whether the dragonfly will exist in the future, but whether the richness, the diversity, the ecological functionality that makes the dragonfly lineage what it is, 300 million years of accumulated biological solution finding, will survive. Whether the globe skimmer will still cross the Indian Ocean in sufficient numbers for the circuit to function. Whether the diverse communities of freshwater dragonfly species that structure aquatic ecosystems around the world will persist in recognizable form. These questions do not have certain answers. They are questions that will be answered by choices, not evolutionary choices, not geological choices, not the slow, impersonal processes of natural selection, but human choices. Choices about how we manage the freshwater resources that sustain not just dragonflies, but the entire fabric of aquatic biological diversity. Choices about how we design our agricultural landscapes, our urban environments, our water infrastructure. Choices about whether the small pond in a garden, the ditch along a country road, the restored gravel pit on the edge of town is worth maintaining, protecting, and allowing to be what it has always been, a habitat. The dragonfly's story connects, finally, to a question that runs beneath all of natural history. What is the relationship between complexity and age? Is there something about the dragonfly that 300 million years of existence has made it more than it would be if it were merely a modern invention? Is there accumulated meaning in that duration, something that we lose if the lineage diminishes or fragments? There is no scientific answer to this question. Science can measure the diversity, the abundance, the ecological function, the genetic variation, all of the quantifiable attributes of a species that tell us about its biological status. But the meaning, the significance, the question of what it matters that this particular lineage has persisted from the Carboniferous to the present, these are questions that science can inform but cannot resolve. What we can say is this: when the dragonfly hovers over a pond, it is doing something that has been done continuously for 300 million years. The pond it hovers over is part of a network of freshwater habitats that is itself 300 million years old in its essential structure, the same kind of habitat that the first dragonfly ancestors exploited, transformed and reformed by plate tectonics and climate change, but continuous in its essential function. The nymph in the sediment below is deploying a hydraulic weapon that is fundamentally unchanged from the weapon deployed by Permian dragonfly ancestors in Permian freshwater ecosystems. The eye that watches the surface from below is constructed on the same optical plan as the eyes that watch the same surface from Triassic nymphs, Jurassic nymphs, Cretaceous nymphs. There is a continuity here that is unlike almost anything else in the biological world. Not the continuity of individual organisms, which live and die in spans measured in months to years. Not the continuity of populations, which persist for thousands to tens of thousands of years before extinction or transformation. But the continuity of a design, a biological solution so comprehensive and so appropriate to its environment that it has been copied, generation after generation, for 300 million years, accumulated from individuals who each lived briefly and died, into a lineage that carries the imprint of the Carboniferous into the modern world. We are lucky to share a planet with such a thing. We are fortunate that this window into deep time still exists, still operates, still hunts and mates and deposits eggs in the ponds and streams of a world that is, in its freshwater places, still recognizable as the world the dragonfly first evolved to inhabit. And we have, in the choices we make about how we manage that freshwater world, the capacity to either honor that 300 million year continuity or to disrupt it in ways that no previous crisis has managed to accomplish. The science of the dragonfly continues to develop in directions that were unimaginable even a generation ago. Genomic studies are revealing the phylogenetic relationships among odonatan lineages with unprecedented precision, allowing the evolutionary history of the order to be reconstructed in detail that the fossil record alone cannot provide. Population genetic studies are mapping the connectivity among populations across landscapes, identifying the corridors through which gene flow moves and the barriers that isolate populations, providing the information needed to design conservation strategies that maintain viable metapopulation networks. Remote sensing technologies, particularly the application of environmental DNA or eDNA analysis to freshwater monitoring, are revolutionizing our ability to detect and census aquatic species, including dragonfly nymphs, without the need for extensive physical sampling. eDNA analysis detects the genetic material shed by organisms into their environment, in this case, DNA fragments released from nymph bodies into the water column, and can identify the species present in a water body from a simple water sample analyzed in a laboratory. This technology has the potential to make freshwater biodiversity monitoring faster, cheaper, and more comprehensive than traditional survey methods, and ongoing studies are validating eDNA approaches for dragonfly monitoring against the traditional methods of physical nymph collection and exuvia surveys. Thermal imaging technology has opened new windows into dragonfly thermoregulation, the behavioral and physiological mechanisms by which these ectothermic animals maintain body temperatures that allow effective flight and hunting even in variable ambient conditions. Dragonflies are not simply passive responders to ambient temperature. They are active thermal managers, exploiting solar radiation, behavioral posturing, and the generation of metabolic heat by warming to maintain thoracic temperatures within a range optimal for flight and predation. The details of how different species manage their thermal environment in different climatic contexts is an active area of research with relevance for understanding how dragonfly species will respond to climate warming. High-resolution tracking technology, miniaturized radio transmitters, harmonic radar tags, and most recently, integrated GPS radio transmitters small enough to carry on a dragonfly, is beginning to provide continuous tracking data on the movements of individual dragonflies through landscapes at scales and resolutions not previously possible. These technologies are revealing the daily and seasonal movement patterns of individual animals, the fidelity of individuals to particular territories or habitats, and the dispersal movements that connect breeding populations across landscapes. The data emerging from these tracking studies are considerably more complex than the simple models of dragonfly movement that were previously available, revealing that individual variation in movement behavior is substantial and ecologically important. The dragonfly, in the end, is many things simultaneously. It is a biological document, a living record of 300 million years of evolutionary history, carrying in its genome the cumulative product of an incomprehensible number of generations of selection. It is an ecological engineer, a keystone predator that structures the communities of freshwater ecosystems around the world, maintaining the balances and interactions that allow these systems to function. It is a biogeographic phenomenon, a global traveler whose migrations connect distant freshwater habitats across ocean basins, maintaining the genetic coherence of globally distributed populations. It is a sensory marvel, an animal that dedicates the majority of its neural tissue to visual processing, executing computations of trajectory and refraction and target detection with a precision and efficiency that continues to inform engineering and neuroscience. But beyond all of these scientific descriptions, as true and as important as they are, the dragonfly is something simpler and more immediate. It is the shape at the water's edge. It is the flash of color against the reed beds. It is the shadow that flickers under the lily pads, the motionless watcher in the sediment, the aerial pursuit that resolves in an invisible fraction of a second. It is the creature that children chase around the margins of ponds on summer afternoons, and the creature that scientists spend careers trying to understand, and the creature that appears in the poetry of cultures that have never heard of each other, because something about its beauty and its movement and its impossible competence in the air moves something deep in human perception. It has been here for 300 million years. It has watched the world change in ways that make every human crisis look like a brief interruption. And it is here now, in the freshwater systems that sustain so much of the planet's biological diversity, performing the same ancient choreography of predation and reproduction that it has performed since before anything recognizable as a modern ecosystem existed. The nymph in the dark water does not know that it is ancient. It does not know that its hydraulic weapon is a technological marvel. It does not know that its brain is performing calculations that human engineers labored for decades to replicate in silicon. It does not know that its wings, when it eventually grows them, will carry it into the same summer sky that dragonfly wings have cut through for 300 million years. It knows only what its extraordinarily refined nervous system tells it, that there is prey ahead at a calculable distance and a predictable future position, and that the labium is ready and that the moment is almost here. And then the moment arrives and the labium strikes and the prey is seized and the ancient cycle continues exactly as it has always continued, underwater, unseen, unhurried, inexorable. This is the dragonfly. This is the story that begins in the Carboniferous and continues, unbroken, to this afternoon, to this pond, to this surface where the light plays in patterns that the nymph below is reading with a precision that 300 million years of natural selection has made absolute. We are fortunate to live in the same world as this creature. We would be fortunate to ensure that the world it inhabits, the freshwater world, the wetland world, the quiet and essential and irreplaceable world of ponds and streams and marshes, persists in the health and richness it has maintained for an unimaginable length of time. The dragonfly has kept faith with the water for 300 million years. The water now needs us to keep faith with it. There are creatures on this planet so perfectly engineered by time itself that nature saw no reason to change them. While entire dynasties of animals rose, dominated, and vanished into the fossil record, while continents drifted and oceans swallowed coastlines whole, while the atmosphere itself transformed beyond recognition, one creature held its form, held its purpose, and held its ground, not through aggression, not through size, not through the blunt force of dominance, but through something far more extraordinary, through absolute, unwavering biological perfection. The dragonfly has been here for 300 million years. Let that number settle for a moment. 300 million years ago, the world was so alien in its composition that you would not recognize it. The forests were not forests as we understand them. The seas held creatures with no surviving relatives. The sky was thick with oxygen, and the insects that flew through it were enormous by any modern measure. And yet, cutting through that ancient air, hunting with precision, hovering with impossible grace, was something that would look immediately familiar to any person alive today. Something with 4 wings arranged in a crosshatch pattern, a long segmented abdomen catching the light, and 2 enormous compound eyes that missed almost nothing. What you are about to discover across the next 2 hours is not simply the story of an insect. It is the story of survival itself. It is the story of a biological system so carefully constructed that time, the great destroyer of almost everything, has chosen, repeatedly and consistently, to leave it alone. This is the story of an animal that outlasted the dinosaurs, that navigated 5 separate mass extinction events, that conquered every freshwater habitat on Earth, and then, in some cases, crossed entire oceans in a single migration cycle. But more than any of that, this is the story of the part of the dragonfly that most people never think about, never see, never consider, the part that is not the shimmering adult hovering over a summer pond, but the slow, ancient, predatory shadow lurking beneath the water's surface. This is the story of the nymph, the creature in the dark, the apex predator of the hidden world below. Please let us know in the comments where you are watching this video and what time you are watching it. And be sure to subscribe to the channel for future videos. To understand what the dragonfly is today, you have to understand what the world was when it first appeared. You have to go back to an era so distant that the very arrangement of the continents was different. You have to go back to the Carboniferous period, roughly 320 million years ago, to a world so lush and oxygen-rich that it barely resembles the planet we inhabit now. The Carboniferous was named for carbon, specifically for the vast carbon deposits, the coal seams that we still extract today, that were formed during this period from the accumulated mass of enormous forests. These were not forests in any sense you would recognize from a walk through the woods today. The dominant plants were not the flowering trees or the conifers of our modern landscape. They were giant club mosses, some stretching 30 meters into the sky. There were enormous horsetails and towering ferns. There were primitive seed plants just beginning their long march toward dominance. And over all of this vegetation, over the swamps and the shallow inland seas and the vast equatorial forests, an atmosphere rich in oxygen, somewhere between 25 and 35% oxygen by volume, compared to the roughly 21% that sustains life today, allowed biology to experiment at an extraordinary scale. Higher oxygen concentration does something remarkable to arthropod physiology. Insects breathe through a system of tubes called tracheae, which deliver oxygen directly to tissues. In a higher oxygen atmosphere, those tubes can be wider, the insect can be larger, and the limits of what arthropod biology can achieve expand dramatically. The result, during the Carboniferous, was a world of giant insects unlike anything that exists today. There were millipede-like creatures stretching nearly 3 meters in length. There were scorpions that dwarfed their modern descendants. And there were dragonfly ancestors, the griffinflies, members of the order Meganisoptera, with wingspans that could exceed 70 centimeters. 70 centimeters, the wingspan of a modern hawk. These creatures, Meganeura and its relatives, were the aerial predators of their age. They had no competition in the sky. The first vertebrates capable of flight were still more than 100 million years in the future. The griffin flies were apex aerial hunters in an ecosystem unlike any that has existed since. And while they were not exactly the dragonflies we recognize today, they were close enough to make the family resemblance unmistakable. The body plan was there. The wing venation pattern, that extraordinary network of veins that gives the dragonfly wing its characteristic lace-like appearance, was there in its essential form. The enormous compound eyes were there. The predatory orientation was there. What this tells us is something profound. The basic dragonfly body plan was not something that evolved slowly over millions of years through gradual, incremental improvement. It appeared relatively rapidly in geological terms as a solution, a complete, effective, elegantly balanced solution to the problem of being a flying predator in a world full of prey. And because it was such a complete solution, because it worked so extraordinarily well, natural selection had very little to improve upon. The organism that we call a dragonfly today is not the product of 300 million years of dramatic evolutionary change. It is the product of 300 million years of almost no change at all. This phenomenon, known to biologists as evolutionary stasis, is one of the most fascinating and poorly understood processes in all of natural history. Most people have an intuitive sense that evolution is a process of constant change, that species are always shifting, always adapting, always becoming something new. And in many cases, that is exactly what happens, but stasis is equally real. When an organism finds a configuration that is profoundly suited to its environment, when it occupies an ecological niche that remains stable across vast stretches of time, natural selection becomes a conservative force rather than an innovative one. It does not push the organism toward new forms. It punishes any deviation from the successful blueprint. The variants that differ from the optimum are eliminated. The successful design persists. The dragonfly is perhaps the most perfect example of evolutionary stasis in the animal kingdom. Looking at fossils from the Jurassic period, some 250 million years after the first dragonfly ancestors appear in the record, you see an animal that is functionally identical to the one you might observe hovering over a pond this afternoon. The wing structure is the same. The body proportions are the same. The essential biological architecture is the same. 250 million years of geological time, and the dragonfly essentially did not move. But stasis is not passivity. The dragonfly has not survived by standing still in a world that also stood still. The world changed dramatically, repeatedly, and catastrophically around this animal, and the dragonfly survived every transformation. Understanding how it did that requires understanding not just the adult dragonfly, which is the form most familiar to human observers, but the entire life cycle, and especially the half of that life cycle that unfolds entirely beneath the water's surface. The life of a dragonfly does not begin in the air. It begins in water. This is the foundational fact that most people miss entirely. When we think of dragonflies, we think of the shimmering aerial acrobat, the jewel-colored body catching the afternoon sun, the hovering stillness followed by explosive, impossibly fast pursuit. But that phase of the dragonfly's life is brief. Depending on the species, the adult stage lasts anywhere from a few weeks to perhaps 6 months. It is spectacular. It is visible. It captures human attention and has done so throughout human history, appearing in artwork and mythology from ancient Egypt to medieval Japan. But it is not where the dragonfly spends most of its life. Most of the dragonfly's existence is spent as a nymph, submerged in freshwater, and this stage can last anywhere from a few months to as long as 5 or 6 years, depending on the species and the environment. During this time, the nymph undergoes a series of molts, typically between 8 and 17, again depending on the species, progressively growing larger and more developed until it reaches the final instar, the last nymphal stage, before the remarkable process of emergence that transforms it into the adult form. The nymph is not a passive, waiting creature. It is not simply a larval stage, a biological holding pattern, a preliminary sketch waiting to become the real thing. The nymph is a fully realized predator in its own right, one of the most effective ambush hunters in any freshwater ecosystem. Understanding the nymph requires setting aside every assumption about what a small aquatic creature can do, because what the dragonfly nymph does is consistently and genuinely astonishing. The freshwater environments that dragonfly nymphs inhabit are extraordinarily diverse. They are found in still ponds and slow-moving rivers. They live in the margins of lakes where aquatic vegetation provides both cover and hunting grounds. Some species prefer fast-moving, oxygen-rich streams. Others have adapted to temporary pools that exist only seasonally. There are species that burrow into the sediment at the bottom of water bodies, remaining invisible from above and below simultaneously. There are species that cling to submerged vegetation, motionless for hours, waiting. There are species that live among leaf litter in forest streams, camouflaged against the substrate so effectively that they are virtually impossible to detect without careful, deliberate searching. What connects all of these diverse nymphs across all of these diverse environments is their role in the ecosystem. The dragonfly nymph is what ecologists call a keystone predator, a species whose presence and activity has effects on the ecosystem that far exceed what you might expect from its size or abundance alone. Keystone species are the structural pillars of their communities. Remove them, and the community does not simply lose one component, it collapses into a different shape entirely. The dragonfly nymph plays this role in freshwater ecosystems around the world, regulating populations of smaller invertebrates, influencing the distribution of aquatic insects, and in many environments serving as a critical link in the food web between the microscopic world of plankton and bacteria and the larger world of fish, birds, and amphibians. To appreciate why the nymph holds this role, you need to understand its hunting strategy. And to understand the hunting strategy, you need to understand the weapon. The labium is the word for the mouthpart that makes the dragonfly nymph one of the most effective predators in the freshwater world. Every insect has a labium. It is a standard component of the insect mouth, a lower lip of sorts, involved in the manipulation and handling of food. In most insects, it is a modest, relatively simple structure. In the dragonfly nymph, evolution has transformed it into something so specialized, so extreme in its modification, that entomologists, when they first fully described it, had difficulty convincing colleagues that such a mechanism was possible in an animal so small. The labium of the dragonfly nymph is a folded, extendable arm. At rest, it is folded beneath the nymph's head, tucked against the underside of the face in a position that completely conceals it. An observer watching a stationary nymph would see no obvious sign of a specialized hunting apparatus. The nymph simply sits, apparently inert. This concealment is itself a crucial part of the hunting strategy, because prey animals that might otherwise flee have no visual cue to trigger their escape response. But when prey comes within range, everything changes in an instant, and the word instant here is not figurative. The mechanism by which the labium strikes involves hydraulic pressure. The nymph rapidly contracts muscles in its abdomen, forcing hemolymph, the insect equivalent of blood, forward through the body and into the labium at high pressure. This sudden hydraulic surge extends the labium outward at a speed that is almost incomprehensible for a biological structure. The entire strike, from the beginning of the extension to the moment of contact with prey, takes approximately 25 milliseconds, 25 thousandths of a second. To put that in perspective, the human eye takes roughly 150 milliseconds to complete a single blink. By the time a human observer's eye could begin to close, the dragonfly nymph's labium could have struck, seized prey, and begun to retract nearly 6 times over. No human observer watching a dragonfly nymph in real time has ever seen the labium strike. It is simply invisible to the unaided eye. High-speed cameras shooting at thousands of frames per second have revealed what this strike actually looks like, and the footage is genuinely startling. The labium unfolds in what appears to be a continuous fluid motion. The prey is seized by the grasping structures at the tip, called the palps, and the entire assembly retracts before most other organisms have had time to begin registering that something has happened. At the tip of the labium, the palps can be structured in different ways depending on the species. In some nymphs, the palps are broad, flattened structures that work like a scoop, engulfing prey. In others, they bear sharp teeth capable of puncturing and gripping struggling prey. In still others, they are long and slender, reaching into crevices to extract hidden invertebrates. The variation in palp structure across the thousands of known dragonfly and damselfly species reflects the remarkable ecological diversity of these animals, each population fine-tuned over generations to its particular prey base and hunting environment. The reach of the labium is also extraordinary relative to the nymph's body size. In many species, a fully extended labium can reach approximately 1/3 of the nymph's body length in front of its face. For a nymph of, say, 3 centimeters in length, this means the labium can reach a centimeter or more beyond the apparent boundary of the animal's body. This extended reach, combined with the invisibility of the weapon at rest, means that prey animals which believe they are at a safe distance, that they have maintained sufficient space from this motionless, apparently passive creature, are in fact well within the strike zone. Many prey animals that have evolved distance-based escape strategies are systematically defeated by this deception. The combined effect of concealment, speed, and reach is a success rate that is extraordinary by any biological standard. Studies of dragonfly nymph predation have documented capture success rates hovering around 95%. This is not a rough estimate or an impressionistic figure. It is a measured, replicated, scientifically established value. The dragonfly nymph, when it strikes, almost always makes contact with its prey. For comparison, a lion, one of the most celebrated apex predators on land, achieves a hunting success rate of roughly 20 to 30%. A cheetah, another iconic predator famous for its speed and precision, succeeds perhaps 40 to 50% of the time under optimal conditions. The dragonfly nymph, moving its hydraulic arm through water in a fraction of a second, succeeds 95 times out of 100. This extraordinary efficiency has a reason beyond the mechanical perfection of the weapon itself. It connects to something even more remarkable about the dragonfly nymph, the way it perceives and processes its world. The dragonfly nymph has a brain that is, by any objective measure, very small. The nymph is a small animal. Its nervous system is compact, its neural tissue occupying a fraction of the space available to larger predators. If you were designing a predator capable of performing complex real-time calculations about the position and trajectory of moving prey, you would not begin with this as your computational substrate. And yet, the dragonfly nymph performs exactly these calculations and performs them with stunning accuracy. To understand what the nymph is actually computing, consider the physical reality of its hunting situation. Many dragonfly nymphs do not restrict their hunting to prey that is entirely submerged with them in the water. Some species, particularly those that position themselves near the water's surface, hunt prey that is above the water, land-based insects, small organisms moving at the water's edge, even small invertebrates resting on the water's surface film. And here the nymph encounters a problem that is not trivial. In fact, it is a problem that human engineers designing underwater targeting systems for submarines must account for using sophisticated mathematics. The problem is refraction. When light passes from one medium to another, say, from air into water, its direction changes. The angle at which it bends depends on the relative densities of the two media and the angle at which it strikes the boundary between them. This is why a stick inserted into water appears to bend at the water's surface. It is why pools appear shallower than they actually are. It is why a fish viewed from above appears to be in a different position than it actually occupies. The light reaching your eye from the submerged fish has been bent by refraction at the water's surface, and what you see is an optical displacement from the fish's true position. This is precisely the problem the dragonfly nymph faces when hunting prey above the water surface. The light reaching the nymph's eyes from a surface level or above surface prey item has been refracted as it passed through the air-water interface. The apparent position of the prey, what the nymph's eyes directly register, is not the prey's actual position. If the nymph simply aimed its labium strike at the apparent position, it would miss consistently. The prey is not there. It is displaced from the apparent position by a predictable amount that depends on geometry, on the angle between the nymph's line of sight and the water surface. What the dragonfly nymph does, and this has been established through careful experimental work, by researchers who systematically manipulated viewing angles and prey positions and measured the accuracy of the nymph's strikes, is apply a correction. The nymph compensates for refraction. It does not strike at the apparent position of the prey. It strikes at the calculated actual position, accounting for the bending of light at the interface between two optical media. This is a calculation. It requires the nymph to register the apparent position of the prey, to determine the angle of its own line of sight relative to the water's surface, and to compute the correction factor needed to identify the true position. In a human engineer, this calculation involves trigonometry. In the dragonfly nymph, it happens automatically, neurally in the time between the detection of prey and the execution of the strike, which you will recall takes 25 milliseconds. The fact that this correction happens has been demonstrated experimentally by presenting dragonfly nymphs with prey targets at various angles through the water surface and confirming that the strike consistently lands at the actual rather than the apparent position of the prey. When researchers set up conditions in which refraction was artificially eliminated by matching the optical density of the medium above the surface to that of the water below, the nymphs initially overcompensated, striking as if correcting for a refraction that was no longer present. This is precisely the behavior you would expect from an animal that has automatically applied a correction factor, and that correction factor, built into the neural programming of the nymph through millions of generations of selection, is now so fundamental to the strike mechanism that it operates whether or not the correction is actually needed. The nymph is, in the most literal sense, doing physics. Not consciously, not deliberately, not with anything resembling human mathematical reasoning, but the computation is real, it is accurate, and it is built into the structure of the nervous system by evolutionary selection pressures that rewarded accuracy and punished misses over an incomprehensible number of generations. But the refraction correction is not the only complex computation happening in the nymph's small brain. The nymph also accounts for the motion of its prey. It does not simply identify where the prey is at the moment the strike is initiated. It predicts where the prey will be at the moment the strike arrives. This is a genuinely sophisticated predictive computation. It requires the nymph to measure the current position of the prey, estimate its velocity and direction of movement, calculate the time the labium will take to reach the target, and identify the future position of the prey at the moment of contact. This kind of predictive targeting, sometimes called interception behavior in the animal behavior literature, is not unique to dragonflies. It is found in other fast-striking predators, including some fish and some frogs. But in the dragonfly, both as nymph and as adult, it is extraordinarily well developed and extraordinarily well studied. The adult dragonfly, in fact, has become one of the most intensively studied models of predictive pursuit behavior in the entire animal behavior literature, precisely because it is so remarkably accurate and because it can be studied in detail using high-speed video and neural recording techniques. The key insight that has emerged from this research is that the dragonfly is not following its prey. It is not tracking the target's position and continuously adjusting its course to stay on the same heading as the target. Instead, it is projecting the future position of the target and moving directly toward that projected interception point. This is geometrically more efficient than pursuit tracking, and it means that the dragonfly reaches the interception point by the shortest possible route rather than by following the longer, curving path that the prey actually takes. The neural mechanisms underlying this computation, located in a remarkably small number of specialized neurons in the dragonfly's visual system, have been mapped with considerable precision, and they represent some of the most elegant biological computing circuits that neuroscience has identified. To fully appreciate the sensory world of the dragonfly both as nymph and as adult, you need to spend some time with the eyes. The eyes are in many ways the defining feature of this animal. Adult dragonflies dedicate approximately 80% of their brain tissue to the processing of visual information. 80%. In a human brain, visual processing occupies a significant fraction of the cortex, but it competes with motor control, language, memory, emotional processing, and the enormous cognitive overhead of social intelligence. In the dragonfly, there is no such competition. Vision is everything. The world for a dragonfly is almost entirely a visual experience, and the investment of neural tissue that supports this visual experience is staggering in its proportions. The compound eye of the adult dragonfly is the most sophisticated compound eye known in the animal kingdom. Unlike the eyes of many other insects, which are roughly hemispherical and provide wide-field but relatively low-resolution vision, the dragonfly's eyes are enormous, wrapping around the head in some species to provide nearly 360 degrees of coverage. In many species, the two compound eyes meet at the top of the head, eliminating almost every blind spot that might allow prey to approach undetected. A dragonfly in flight knows what is happening in virtually every direction simultaneously. Within this enormous visual field, different regions of the eye provide different kinds of information. The frontal dorsal region, the forward upper quadrant of the eye, the zone that looks forward and slightly upward, contains the highest density of photoreceptors. This is the acute zone, the region of maximum resolution, and it is positioned to view the sky. This is not accidental. Dragonflies hunt by looking upward, against the bright background of the sky, where prey items appear as dark silhouettes. The acute zone is optimized for exactly this viewing geometry, and it contains specialized photoreceptors tuned to maximize the contrast between a small dark object and a bright background. The result is visual acuity that is, by some measures, the best in the insect world. Dragonflies can detect prey items at distances of up to 10 meters. They can track individual insect visual noise of a complex background, a moving target against the irregular pattern of sunlit vegetation, for instance, with a consistency that imaging scientists find genuinely impressive. And because the acute zone subtends a relatively narrow angle, the dragonfly must orient its head to bring the target into this high-resolution region, which it does through rapid, precise head movements that can track a moving prey item with remarkable fidelity. The dragonfly also has an extraordinary ability to filter out visual information that is irrelevant to its hunting goals. Neurons in the dragonfly's visual system are selective for small, moving objects against complex backgrounds. They respond vigorously to exactly the kind of visual signature that a small flying insect produces, and they are relatively unresponsive to background motion caused by wind moving through vegetation or the dragonfly's own movement through space. This selective filtering means that the dragonfly's visual system performs something analogous to the background subtraction algorithms used in machine vision systems. It strips away the irrelevant and highlights the target, delivering a clear hunting signal even in a visually cluttered environment. The nymph's visual system is different from the adult's. It is adapted for the murkier, more light-limited environment of the underwater world, but it shares the fundamental orientation toward visual precision. The nymph's eyes are large relative to its head size, forward-facing enough to provide genuine binocular vision in the frontal field, which is critical for the depth perception needed to accurately place the labium strike, and sensitive to the specific light conditions prevalent in the freshwater environments the nymph inhabits. In some deep water or cave-dwelling nymph species, the eyes are reduced and other sensory systems, primarily mechanosensory systems that detect water movement, are enhanced. But in the vast majority of nymph species, vision is the primary sense, and the eyes are the organs around which hunting behavior is organized. The integration with the hydraulic strike mechanism, the refraction correction, and the predictive trajectory calculation represents a suite of adaptations that take in dragonfly nymph, one of the most engineered predators in the biological world. Each component reinforces the other. The system feeds accurate into the trajectory. The trajectory calculation places the refraction-corrected target position in the future rather than the present. The hydraulic mechanism delivers the labium to that future position with a speed that gives the prey no time to respond. And the concealment of the weapon at rest ensures that the prey never triggers its escape response before the strike is already complete. It is, in the most literal sense, a system, and systems of this kind, integrated, mutually reinforcing, with each component optimized in relation to all the others, are not assembled quickly. They are the product of vast evolutionary time, of selection pressure applied across millions of generations, iteratively refining each component and the interactions between components. The 300 million year history of the dragonfly is not just a timeline of survival. It is the duration of a design process, and the product of that process is something that operates at a level of engineering sophistication that continues to surprise researchers who study it. Having established what the dragonfly nymph is capable of in terms of predation, it is worth spending time on what it actually eats and what eats it, because both of these relationships are central to understanding the nymph's role in the freshwater ecosystem. The dragonfly nymph is an opportunistic generalist in its diet. It will eat essentially anything it can physically subdue that comes within reach of its labium. In the early instars, the youngest, smallest nymphs, the prey base consists primarily of microscopic organisms, water fleas, copepods, small aquatic worms, midge larvae. As the nymph progresses through its molts and grows larger, the prey base expands dramatically. Medium-sized nymphs take mosquito larvae, small mayfly nymphs, chironomid larvae, small water beetles, amphipods, and aquatic snails. Larger nymphs in the final instars, which can be quite substantial animals, up to 5 or 6 centimeters in length in some species, are capable of tackling prey that most observers find genuinely shocking for an insect predator. Large dragonfly nymphs eat tadpoles. They eat small fish. They eat the larvae of other large insects, including the larvae of other dragonfly species. In competitive or resource-limited environments, cannibalism of smaller conspecifics is documented in many species. And the same predictive, hydraulic, high-precision strike mechanism that works so effectively on tiny invertebrates scales perfectly to these larger prey items. A 5-centimeter dragonfly nymph striking at a small fish is deploying the same weapon, the same targeting system, the same hydraulic mechanism, but calibrated to a larger target and achieving the same extraordinary success rate. This size range in prey, from microscopic plankton to small vertebrates, is one of the reasons the dragonfly nymph functions so effectively as a keystone predator. It is not locked into a narrow ecological role, exploiting a single prey type and thus vulnerable to the boom and bust cycles that can destabilize specialist predators. Instead, it spans multiple trophic levels simultaneously, eating across a broad range of prey sizes and types, and this dietary breadth gives it resilience that specialist predators lack. At the same time, the nymph itself is prey for a variety of larger predators, and this predation pressure has shaped many aspects of nymph behavior and morphology. Fish are the primary predators of dragonfly nymphs in most aquatic environments. Herons and kingfishers take nymphs in shallow water. Larger dragonfly nymphs eat smaller dragonfly nymphs, as already noted.